Welcome to this lecture on hormones. The goal of today will be to understand how hormones work and there's some of the receptors. So let's start with a case study. 18-month-old boy was bitten on his right hand early in the morning when he was playing outside on the floor just in front of his parents' house. He presented at the clinic less than 1 hour after the bite. After the examination, there was no bleeding, no neurological abnormalities, but the hand and the right forearm were swollen and extremely painful. Here's a picture of the initial examination. What's your diagnosis? The diagnosis was snake bite, and because of the extensive swelling, the child immediately received his first dose of antivenom and some antibiotics and some vaccinations, including tetanus, and some ibuprofen helped the swelling, but that is what happened eventually. He lost all of his skin, and we'll talk about why that is, and we'll eventually just discuss his recovery. So today we're talking about hormones and chemical messengers. A hormone is a long-distance chemical signal that travels in the blood or the lymph. Autocrine signals are chemicals that exert effects in the same cell that it secretes. Paracrines are locally acting chemicals in the region, but that they're not associated with that direct cell that they send out signals. So that we want to distinguish what these messengers are so that we can get a good idea of how they're acting. So hormones act in a couple different ways. One, they alter plasma membrane permeability of the membrane potential, or by opening and closing. I said that wrong. They alter the plasma membrane permeability by opening or closing ion channels. They stimulate synthesis of proteins or regulatory molecules. We'll show you that mechanism. They activate or deactivate an enzyme systems. They induce secretory activity, or they stimulate mitosis. And we'll talk about all of these mechanisms generally and how they're effective. So two general mechanisms of hormone action. We have water-soluble hormones. These are all the amino acid-based hormones except the thyroid hormone. They cannot enter the target cell. They act on a plasma membrane receptor. Usually these are G-protein, GPCRs, as we're calling it, G-protein coupled receptors. They then are inducing an intracellular second messenger signal that mediates the target cell's response. We'll talk about this in a few days when we talk about biosignaling. Otherwise, we have lipid-soluble hormones. They're both steroid and thyroid hormones. They act on an intracellular receptor that directly activates genes. These hormones can permeate lipid membranes because they are lipid soluble, and by that mechanism then they can initiate. Usually it's transcription. Some terms. Neuron is a nerve cell. Neurotransmitter is a chemical messenger between a neuron and another target cell. Another neuron, a muscle, or a cell of a gland. Hormone, again, is the chemical messenger, and the distinction between a neurotransmitter and a hormone is physiological. It's not chemical, so it depends on whether the molecule acts over a short distance across the synapse or across a long distance through the blood, or where it takes it to its site of action. So it's basically just a local distinction. There are three types of molecules for communications that are necessary. We need a receptor. That's the protein that binds the ligand or the hormone or whatever messenger we have. We have chemical messengers. Those are also referred to as ligands. They interact with the receptor, usually in a very lock and key type model. That's very reminiscent of the enzymes. And we also have second messengers, in which a messenger from the receptor is sent inside the cell and it is amplified, and it amplifies the signal usually for transcription. So there's 5 types of chemical messengers. We have steroids. The steroid hormone is, and we'll talk about each of these. Steroid hormone, an amino acid. We have peptidergic. This is the insulin type. We have androgenic. This is an acetylcholine type. And it's related, I guess I should say, where we have epinephrine, and then we have cholinergic, where we have acetylcholine type receptors. We'll talk about each one of these, but there's 5 classes. You need to know all of them. So the first kind is the acetylcholinergic receptor. I have struggled with that word every time I say it. Acetylcholine is sent across a nerve synapse in order for the message to occur. You can see the structure of acetylcholine. It's pretty similar to most small molecules. Acetylcholine is often it is abbreviated ACH. It's the main cholinergic messenger. Here's an example, a 3D structure of one. You can see this, the top view on the left. You can see acetylcholine is the red part of the binding molecules. It's the ligand right there. On the right, you see it from the side view. The gray part is where the membrane is. The top part is the extracellular, and on the bottom is the intracellular membrane. I would encourage you to look at this in PyMol if you have an opportunity. Acetylcholine, again, it's an ion-gated channel receptor. It's going to open up, and you're going to have things like sodium, potassium pumps that are going to open up based on the reception of acetylcholine. That's going to send nerve signals down it's going to send basically nerve cells down the axon. Next kind is amino acid messengers. Amino acid messengers, some examples, some excitatory examples are glutamate, aspartate, and cysteine. Other inhibitory neurotransmitters include glycine and taurine, beta-alanine, and GABA. And so these are all pretty normal amino acid messengers that are used to send messages. So next example is adrenergic messengers, and we have a receptor on the membrane. Again, this is probably GPCR. The GPCR is going to engage in the G protein coupled receptor or protein. The G protein is going to separate. That's going to cause cyclic AMP to be translocated and put onto a protein kinase. The protein kinase is going to phosphorylate something, and it's going to open up an ion channel. So that's how the general mechanism. Again, once the monoamine binds, you're going to have the primary messenger or neurotransmitter or hormone that's going to bind. We have our cyclic AMP that's going to then send the signal. We're going to open up some kind of ion channel. Peptidergic messengers, usually this is something like insulin. This is the insulin, something like GLUT4, where you're going to have insulin bound to that molecule, and then we have some kind of second messenger system. Usually it's associated with a phosphorylation event, an autophosphorylation. You have tyrosine kinases domain. They're going to autophosphorylate. That's going to send down a second messenger. And again, we'll cover this in biosignaling quite extensively. In fact, we'll probably talk about specifically this GLUT4 mechanism as it goes down. And so that there's an intracellular insulin effects based on the receptor, on the insulin receptor. Another peptidergic messenger is a glucagon. Here's an example of how all of it, there's a receptor binding, it stimulates a G protein. We have adenylate cyclase that's going to phosphorylate cyclic AMP. Cyclic AMP increases activity of phosphofructose kinase 2 all the way down to these activities when we get suppressed glycolysis or increased increased gluconeogenesis. That's the kind of how these kind of things kind of function. Next we have steroid hormones. Steroid hormones are usually lipophilic, so they'll go right to the exterior cellular membrane. Then they'll bind to a receptor hormone that's in the cytoplasm. That then will go permeate the nuclear membrane. That'll initiate transcription. We get some kind of transcript that gets translated to a protein. We have a new protein that does some kind of work. So that's how steroid messengers generally function. Again, this kind of gives you an idea. We have steroid hormone diffuses the plasma membrane. It binds the intracellular receptor. The receptor then complex enters the nucleus. The receptor binds the complex and it binds to the promoter region as a transcription factor. We get an mRNA transcript that's produced after transcription, and then we get a new protein after translation. That's the general mechanism for a steroid. So steroid sex hormones are also very common. They are synthesized again from cholesterol. Cholesterol goes to progesterone, and progesterone goes to the various forms of estrogen and testosterone, which are shown on the right. So they're responsible for secondary sex characteristics, and the sex hormones, again, also can initiate a translation and So prostaglandin is the last family of chemical messengers. Prostaglandin is often abbreviated PG, or a group of hormones that have a 20-carbon skeleton. They are of prostanoic acid. They have a ton of biological mechanisms and relevance found in almost every tissue in the body, and the slight structural differences between the different prostaglandins give different biological outcomes. In fact, it can be even like opposite in some tissues. They are autocrine and paracrine factors, and generally, so they're relatively close, and they're produced in many places across the body. So there are things like vasodilators. They prevent blood platelet aggregation. They're involved in inflammation. Anyway, they serve all kinds of smooth contraction and regulation of smooth tissue, muscle tissue, contractions in pregnancy. So there's all kinds of different mechanisms of these prostaglandins, and there's a receptor for them. That receptor is a transmembrane, which we'll talk again about. So we're getting a little bit ahead of ourselves with transmembrane, and we usually get into second messenger effects. Chemical messengers are also classified by how they work. So they can activate enzyme, they can affect the synthesis of enzymes, they can affect the permeability of membranes, or they can act directly or through a second messenger. So they can do any number of things. There's all different kinds of mechanisms of action of chemical messengers. Large percentage of drugs we encounter, this is the reason we're talking about this, large percent of the drugs we encounter in medical practice try to influence chemical communications, and this is where drug families come in, and drug type of drugs. An antagonist drug blocks a receptor and prevents its stimulation. An agonist competes with a natural messenger for receptor uptake, therefore it stimulates the receptor. So we also have other drugs that increase the concentration of a messenger by inhibiting its removal from its receptors. Other drugs inhibit or activate specific enzymes inside the cell. So we have all different kinds of mechanisms. These are generally small molecule type discussions, although there are many antagonists, especially, that are antibodies that prevent the stimulation. So let's go back. So all the way to snake bite. So the patient received a 7-day prophylactic antibiotic therapy. For every 3 days, the wound was cleaned and the dressing was replaced. After 11 surgical interventions and 33 days in the hospital, the child's arm was skin grafted with good results. So that's the final result of the snake bite. I know we've gone kind of fast on this hormone. Hopefully you get a general idea. There's not a lot of topic and discussion in this. In fact, in 460 we don't even mention hormones, and biosignaling is almost forgotten in the process. But I think it's very important for the pharmacy side of things, because these are drug targets. So I at least want to at least introduce the concepts to you. We will cover this topic in greater detail when we talk about biosignaling, but I wanted to introduce the concepts. We'll hone in on these concepts again when we talk about biosignaling in a couple days. They're in the lipid family, which is the steroids are, and so that's the reason we're talking about them here. But generally speaking, we're going to discuss them in greater detail when we talk about biosignaling. Look forward to seeing you in the next lecture.